Okay, our next speaker from UC Davis, Dr. Imitias Kandi, is currently is assistant professor of plant reproductive biology in the Department of Plant Science at UC Davis. He also serves as the director of research for the Seed Biotechnology Center. Imitias obtained his PhD in plant development and genetics from the India Institute of Science, Bangalore, India. Before joining plant science, he was a postdoc scholar at the Department of Plant Biology at UC Davis. His research interests focus on the understanding cell fate specification during seed development, as well as the seed dormancy and vigor. Welcome, Imitias. Okay, good.
Morning, everyone. Thank you, Alfred, for the invite and the introduction, and Todd as well. I'm going to tell you about our work, which is mainly done in rice, on synthetic apomixis. And I'll tell you how we can propagate a crop, rice, that has propagated itself through sexual reproduction for about 10,000 years. And we can actually propagate it clonally through seeds. It does. Okay. So the previous speaker made my life very easy, introducing apomixis. So apomixis is an asexual mode of reproduction through seeds, where some plant species are able to clone themselves through seeds. So it's essentially cloning through seeds. We know about apomixis for a long time. The first reported incidence of apomixis is from this plant called native holly. So there was a single plant planted in the UK, introduced it from Australia, and it was able to make seeds on its own. Now you're going to say, what was special about that? Plants do that all the time. But in native holly, the male and female plants are separate. So it was a single female plant without the involvement of males able to do it. In nature, as Peggy said, there are several ways of achieving apomixis. It can be gametophytic, or it can be sporophytic. And it has been found in more than 400 angiospermic species. So it's pretty widespread in nature, but yet it's only found in about 30 families or so. So in that sense, it's quite rare. So why do we care about apomixis so much? One, it's a natural form of reproduction and really interesting, but it also opened up the possibility that we could actually propagate hybrids through apomixis, which really would be a useful technology for farmers or for growers in general. So as you all know, this is nothing new, that we can cross certain specific inbred varieties in different crops, and that results in the enhancement of vigor in the F1 generation. In no other crop has we realized the application of hybrid vigor than in corn. So this slide shows, okay, there's no pointer. So this slide shows the corn production before 1920s. It was quite, still. There was no increase in yield, as because farmers were saving their own seeds. And it's around 1920s, thanks to George Schull, when we learned how we can make F1 hybrids, which have enhanced vigor. And then after that, you can see that there has been an exponential increase in corn yields because of the use of hybrids. And it's because in corn we can relatively easily make these F1 hybrids. We have a simpler reproductive biology. The male and female inflorescences are separate. We can make female plants by just detasseling the plants and then grow male plants interspersed between the female plants, and wind does the rest. So we can relatively easily create F1 seeds in corn. But that's not the case in other cereal crops or major crops. I'll be generally talking about cereals, say for example, rice. And it's true in wheat, barley, sorghum, and others as well. The floral structure is not that simple. So we have male and female reproductive parts housed within the same flower. And then we require sometimes these 3-parent crosses to create these F1 hybrids. So this complex reproductive biology makes actually these F1 hybrid production, even though we have such yield enhancements, it makes it uneconomical from a commercial point of view. So for example, what hybrids, say for example in rice, can do, this slide draws a comparison of yield increases with hybrids between two countries, neighboring countries, India and China. So India uses almost 1.5 times or 50% more land for rice cultivation than China, but it produces only half the amount of rice that China does. And why is China producing so much more? Because not the 100%, but just the 50% of the land in China is under hybrid rice cultivation. So hybrids can really enhance yields. But because of the complexity of making these F1 hybrid seeds and the production costs, we don't use basically F1s for hybrid production in these crops. So we produce F1s, they undergo sexual reproduction, which results in the segregation of these hybrid characteristics and results in the loss of hybrid vigor. But as Peggy was saying, if we could make these F1 hybrids to undergo apomixis, then we do not run into this problem. We can basically produce self-replicating hybrids, and then we won't lose. And that will make basically the economics of these F1 hybrids more viable. So we knew about apomixis, and there were natural or the wild relatives of several crops. What Peggy was saying again, that the easiest way for us to make apomixis work in crops was to just simply integrate it. So this is an example in penicetum or pearl millet, but there are wild relatives in other crops, say for example wheat and maize, and similar experiments were done to basically just integrate the apomictic trait into the crop traits. But what this did was we were able to make basically integrate apomictic trait, but when you are trying to now backcross these hybrids basically back into that crop, in the process we lost this apomictic trait mainly because there was not viable seed production, which again was probably because there was not viable endosperm. So we in the lab did not start out to engineer apomixis in crop plants, but rather address a very fundamental question about reproductive biology, which is what is the gene expression in gametes, and that's egg cells and sperm cells in plants, and how does the gene expression change after fertilization in the zygotes, and how does that affect zygotic development? So this was to basically, at the time, address this huge controversy in Arabidopsis, where two contrasting theories were put forward. how basically zygotic gene expression works. In animals, the initial zygotic development is completely controlled by the gene products, that's proteins and RNAs, that are stored in the egg cell. And the male genome does not participate in the initial zygotic development. But how does that work in plants? So there were these two contrasting theories. One, which was that the male genome, it was activated slowly, and there was a huge maternal bias in the gene expression in plants. But then this other theory suggested that's not the case. The male genome is activated immediately after fertilization, and it fires equally as much as the female genome does. So, but these studies, the initial studies, were not actually conducted in actual zygotes. They were conducted in 2 to 4 cell pro-embryos. I'm not going to get into this controversy anymore, but we thought we could address this question better in rice. Because in rice, it takes only 30 to 40 minutes for fertilization to occur after you have deposited the pollen or after the pollination has happened. And we can dissect single-cell zygotes in rice. It's a pain in the neck because it involves a dissection, a manual dissection, but we could isolate single-cell zygotes. And because the time from pollination to actual fertilization is so short, we can actually time stage these zygotes. So, for example, in rice, plasmogamy happens by 1 hour, and by 2.5 hours after pollination, we have karyogamy. But in Arabidopsis, remember, this is highly variable. So the time from pollination to actual fertilization, so that was the reason why those single-cell studies, those studies were carried out in those pro-embryos. So a long story short here, what we found was that in rice, most of the gene expression in zygotes is highly maternally biased, meaning there is a huge maternal bias for gene expression in zygotes. But in the center here, there are genes which have biparental expression that were not basically expressed in one of the parents, meaning the male genome is activated immediately after the fertilization. But what we also found were these specific, this certain number of genes, a couple of dozen of them, which are in the blue towards the left, which had a paternal expression, meaning they were expressed only from the genome that's inherited from the sperm cells. And among these 2 dozen genes, we found 2 transcription factors, a baby boom gene and a WOX gene, that were expressed only from the male genomes, meaning the genome that you inherit from the sperm cell. This is the SNP analysis that confirms that in these crosses between Japonica and Indica rice, that they are expressed only from one allele, that's the male allele in the zygotes. So we looked a little deeper into baby boom 1 expression, and we found that it is expressed in the sperm cells, so even before fertilization, but it's not present in the egg cells. This shows the same thing. This is a baby boom GFP line showing expression actually in this pollen grain in the 2 sperm cells before the fertilization happens. You have to take my word for the other gene, which is WOX9A. We did the experiments. It's just that for the sake of time, I'm not showing the data. But for WOX9A, it doesn't express either in egg cells or in sperm cells. Its expression turns on de novo after the fertilization. So we did some other experiments. We ectopically overexpressed baby boom gene in rice, and we found that it can induce somatic embryogenesis even on leaves. So it's a gene that's certainly capable of inducing embryogenesis. It doesn't express in egg cells. It expresses in the sperm cells, and after fertilization in the zygotes, it expresses only from the male alleles. So that raises a big question for us. Is it possible that this gene, baby boom 1, is actually an embryogenic trigger after fertilization? So how do you test that is we ask a simple question. If we express it in the egg cells, where it doesn't normally express, would that bypass the need for fertilization and induce what's called parthenogenesis? And when we do that, that's exactly what it does. So if we drive the expression of baby boom 1 gene in rice through an egg cell-specific promoter, it leads to parthenogenesis. That's embryo development without fertilization. So you can do away with the requirement of sperm cell with the expression of this one gene, and you can initiate embryogenesis. If we look at the seeds from these plants, we can obtain haploid plants, obviously, at about 30 to 35%. So what about the other gene, the WOX9A? Does WOX9A have capability to induce parthenogenesis? The answer is no. We screened about more than 1,000 plants from 8 different lines, and there were no haploid plants. But if we express WOX9A on top of baby boom 1, what it does is it increases the parthenogenetic ability of baby boom 1. So, for example, shown here in this graph is that we used one line where the background haploid induction frequency of baby boom 1 by itself was 20%. But if WOX9A is hemizygous, it can bump it up to about 60%. But in a homozygous WOX9A condition, we can increase it to about 98%. So what that tells us is that WOX9A acts synergistically with baby boom 1 in controlling parthenogenesis or embryogenesis in rice. I'm not showing you the data, but both of these genes redundantly with other WOX genes and baby boom with other baby boom genes are redundantly required for embryo initiation, but I'm going to skip that part. So what we learned so far was that I'm not going to, again, get into the dicots or what we know about zygotic development in Arabidopsis, but I'm going to focus on monocots, or those are the studies based in rice, is that baby boom genes are inherited from sperm cells, and in the zygote after fertilization, they induce the expression of other auxin biosynthesis genes. Again, I'm not showing the data, but it's published now. So something called auxin biosynthesis genes or YACA genes, and that's how they activate basically embryo initiation. WOX9A is expressed in the zygotes after fertilization only from the male alleles. If you're going to ask me, do baby boom and WOX9A have a direct genetic regulation? We don't know yet, but we're looking into that. This is how basically what we know about baby boom function in sexual reproduction so far. So to synthesize apomixis in rice, as I said, sexual reproduction involves 2 fundamental processes, which is meiosis and fertilization. And to be able to engineer apomixis in any crop plant, in a sexually reproducing crop plant, you would need to bypass both of these processes. So with the egg cell expression of baby boom 1, we could substitute we could substitute fertilization with parthenogenesis, but there is still the other part of sexual reproduction, which is meiosis. So fortunately for us, our collaborators in France, Raphaël Mercier's group, developed this amazing procedure called MyMay, which stands for mitosis instead of meiosis. So what it does is it's a combination of 3 genes that basically control 3 different steps in meiosis. And you create a triple mutant of these genes, and you can convert meiotic division into a mitotic-like division. So there is no homologous pairing, there is no recombination, and there is no second meiotic division. What you end up having is gametes, which are diploid without segregation and without recombination. So if we combine now MyMay with baby boom-induced parthenogenesis, can we produce clonal plants? So the idea here is MyMay will produce these diploid gametes without segregation and recombination, and then parthenogenesis by baby boom XL expression should lead to 2N clones. But if they undergo sexual reproduction, they won't be actually recombined, but the ploidy will double from MyMay only, and then we will end up having 4N sexual progenies. So we did this, and what we found was that we could recover actually diploid clonal progenies in about same frequency as that of the baby boom parthenogenesis frequency, about 30%. So tetraploids in rice are quite distinct from diploids. They have these long awns, they're sterile, so that made our life easy identifying diploids from tetraploids. This was actually a proof of concept created in an inbred cultivar of rice, Kita-K. But there were two basically bottlenecks to be able to apply this technology actually in the field. One is that would the fixation of heterozygosity actually translate into fixation of heterosis? Because there were some reports indicating that the epigenetics of sperm cell is actually essential for that hybrid vigor. And the second thing is that at this 30 to 35% apomictic frequency, you cannot introduce in the field. It needs to be at least more than 80% to about those agriculturally accept limits. So we collaborated with Emmanuel Guidardoni at CIRAD in France, because we don't have rice hybrids, they had them. So we threw our constructs at this hybrid that's actually grown here, well, not here, in South America, in Brazil, but it was created at CIRAD in France. So it's again created by doing those 3-parent crosses. It's known for its high grain quality and low breakage rate. So when we apply our constructs to this, so what we recovered were F1 hybrids clonally reproducing at about 95% efficiency. And we have some lines which are actually 99% apomictic. But the cool thing here is that you can see that none of the, on the left side of the screen is an F1 hybrid. And when you allow it to undergo sexual reproduction, everything segregates. But when you fix it with apomixis, nothing segregates, everything looks the same. So with this combining MyMay and baby boom parthenogenesis, this apomictic system actually mimics the diplospory form of natural apomixis. And you can actually fix heterosis or hybrid vigor with this technology. So yeah, the fixation of heterozygosity actually translates into fixation of heterosis. We could run them through different generations and nothing segregates, they just look the same. So is this the only way of engineering apomixis in crop plants? No, there are other ways. So what we have used is combine MyMay with parthenogenesis by baby boom 1, but we could essentially combine it with other forms of, which is not, in my opinion, true parthenogenesis, but they mostly result in chromosome elimination after fertilization, say, for example, using genes like matrilineal or SNH3, but we can essentially achieve the same result. But the frequency of apomixis with these other systems is low. Say, for example, with matrilineal, only 90% of the seeds recovered were apomictic, but then there is huge seed sterility with these chromosome elimination procedures as well. And recently, another gene, which is the PAR gene or parthenogenesis gene from dandelion, it was discovered. It's responsible for parthenogenesis in apomictic dandelions. When that was expressed along with MyMay in rice, it could also do apomixis at about 30 to 35%. So that's about it. With that, I would like to thank my group at the top, my colleagues at Sundar Lab, which was where I did my postdoc, our collaborators, Emmanuel and Rafael at CIRAD, and Rafael is at Max Planck Cologne now, and everyone else who fund our research, and thank you all for listening to me.
We have time for 2 or 3 questions. Any?
Very nice talk, thank you. So this would be considered genetically modified, right? Because you have to have the BBM.
Yes.
Right.
Right now, it's a GMO.
Okay. And could you also talk a little bit about the boosted breeding that companies, or how this compares with, and that companies like Ohalo have been talking about, to the extent you're aware of and can talk about it?
Well,
Because they're also trying to, like, essentially, as I understand, make polyploids, right? To get hybrid vigor and heterosis.
so I had a chance to actually read a little bit about Ohalo. So what they're saying is that basically using MyMay to produce diploid gametes, and then, say, for example, converting diploid crops into tetraploid. That's my understanding, and would that boost the yield? And they have shown some results that can happen, but yeah.
Thank you for that talk. That was interesting. I have a question about long-term. Could farmers just propagate this seed then, the way they used to, without having to buy it from a company?
You're getting me into something I don't want to get into. But the short answer is yes.
Very nice presentation, thank you.
Thank you.
I would like to know how you can apply this technology to polyploidy species like strawberry, sugarcane.
It essentially using same thing, but the only question is, would the parthenogenesis genes work, and would the MyMay genes work? So, but there shouldn't be any difference whether it's polyploid or it's diploid, because what Peggy was also saying is baby booms are dominant. So the parthenogenesis dominant, and if you're creating a mutant in MyMay genes, or hopefully in the next gene that Peggy is going to tell, a single gene for apomiosis, it will make it all the more easier.
So if the polyploid species and it have difficulty in selfing, then how you can apply this technology? Because you cannot segregate out by selfing. They have low fertility in selfie. For those plant species, what is your strategy to overcome and to apply this technology?
I really didn't understand your question. Can you
I
repeat
mean,
it,
some polyploid species, they have difficulty in fertility in S1, S2. Then for those species, how you can try to overcome?
Oh, you're talking about self-incompatibility.
Yeah,
Right.
because although you use this technology, but in S1 you cannot get the.
Yeah, I
So
understand
how-
that. So,
how
say, for
you are trying
example,
to overcome,
in
yeah.
some tomato and potato species, right?
And although in some
Yeah.
strawberry
Yeah.
species,
Yeah, I would say maybe knocking out those sRNase genes along with the MyMay genes, probably.
yeah. Thank you.
Thank you. Okay.
Thank you.